The next speaker is Professor Roland Grafström, a colleague of mine from the Karolinska, Professor of Toxicology at the Institute of Environmental Medicine. His academic background is in the development and use of both in silico and also primarily from the beginning in vitro test methods and application to toxicology and risk assessment. He's been present in many EU projects. Some of them I've been there as well, so we've been colleagues, and one of the moment we are colleagues in is the EU Risk Hunt 3R program, which is part of the ASPIS program, which is more chemically orientated again. But and obviously high throughput screening methods are central to a toolbox that might find application in the area of nanosafety in the future. So the talk is entitled EU Safe and Sustainability Projects, Safe and Sustainable by Design Project towards HTS testing. That was a mouthful, Roland. Off we go.
All right, thanks very much, and thank you, Penny, for giving me this title. I'll be very glad to talk briefly on high throughput testing, and also to show photos here of my senior scientist collaborators, Penny, Vesa Hongisto, and Pekka Kohonen in these projects that I'll just quickly review for you. So shown here are our active EU projects, which cover the chemicals and nanomaterial safety areas. And I would say that all of them, to some degree, have now the understanding and acceptance of the safety and sustainability of design. And almost all of them consider high throughput as a concept to quickly generate data and get ideas about a large number of materials or chemicals in terms of their hazard and so forth. So I'll come back a little bit very briefly in this talk to these projects at the end, but wanted rather than to give you instead the overview of a slide that I think many of you have seen many times. And it was actually, considering we talk about time for acceptance and implementation, put together the first time by myself and co-workers almost exactly 13 years ago for Electorate ECHA in Helsinki, so 2008, November, December. Essentially what you hopefully see here, I don't know, maybe the title is hidden there because it is for me, but maybe not for you, is the Karolinska MISFIC high throughput tiered approach to assessment of nanomaterials. And what you can see here is essentially the idea of beginning in a tiered approach with high throughput screening. And that is something I like to underline, and we can come back to in discussion. It isn't standing alone. It's really something that is part of a bigger scenario of understanding hazards of nanomaterials, and certainly NAMs are key here. So the idea is to go, we can have a couple of directions and look at this real briefly, is that you begin with multiple chemicals or nanomaterials in this high throughput initial screen. You move over to other types of methodologies and use more selected concentration based on the understanding that you have at the first tier, and then you go really deep for those which you are really like to study much more in detail, and you come into the areas that we heard in the recent presentation on modeling, and including in this case omics data, and going all the way to mode of action and AOP, etc. And basically the idea is, if we go at the lower tier here in the slide, to go from the traditional cell culture where you have even in a large experiment, maybe a possibility to assess 30 to 100 endpoints, to move to a machine-assisted operational environment where you can do cell-based high throughput screens, and you can go up to quite easily of 25,000 analyses within the same time frame that you would do a classical cell culture experiment. And you would use a couple of endpoints that are considered key to what you're after. And in the next slide, I'll show that in a moment. Then you can have immunochemical assays, or you go to, again, various high throughput omics analyses with diverse technologies like microarray or sequencing, etc. And you can also go then to analyze reduced features, gene sets, for cost basis, and still get really, really far with understanding pretty much completely what goes on within a cell. So, this, I think the most recent version of this is available in the paper that Penny hinted on in her introductory presentations. You can look at Newmark et al. 2020 for details of a busy slide like this. So just wanted to then show that particularly at this work that we do with the cooperation of Karolinska and MISVIC is done at MISVIC, where the setup is available for these kind of assays. And a whole number of different assays are combined. I'll come back to that in the bottom of the slide. In short time points, also longer time points are very important. And essentially it's area under the curve or reversely area under the curve, depending on how you see it from toxicology standpoint, that you assess over broad concentration ranges going at least 3 orders of magnitudes in repeat experiments. And typically if you analyze 20, 30 nanomaterials, you would generate something like 50,000 or 100,000 data points and a broad coverage of toxicologically relevant data. The TOX5 score at MISVIC, so it replaces sort of the one endpoint time point concept with the multi-time endpoint score. Covers growth, death mechanisms, energy state, ATP changes, genotoxicity, reactive oxygen species. Pretty superficial to one degree, but the point here isn't to come to full conclusion. The point is to get the initial scoring, an idea, and ability to analyze these metrics together and get some kind of a ranking, an idea whether materials are dangerous or toxic. And Penny actually was the one who introduced in our group the idea we should use the TOXPI, Toxicological Prioritization Index, which is used by the United States Environmental Protection Agency and National Toxicology Program. So there's a huge amount of data that is also a reference point to work that we do with many, many nanomaterials and certainly hundreds and maybe even thousands of chemicals. It's all integrated. And also to say that it can be coupled. And Penny actually has done excellent work here with physiochemical properties and a huge number. So outside of the biological analysis, it can be combined with other things. So this is actually the way we see this tiered approach, that it fits into a non-driven risk assessment paradigm. And the figure that Penny mentioned or showed in her presentation actually emanates and was inspired by this figure from Adelaide, where we then see that the type of biological assessment that I talk about now function in high content information assay, human cells and model, analyzing dose response. And the later omics part, which I can come back to if anybody likes to, is also handling that aspect, but going into the computational modeling. And then overall, coupled to an AUP and mode of action analysis, we can also include some limited PBK modeling and dosimetry modeling in the experiments. And a very important point is the final sentence there. It all works together very nicely to look at very low levels of toxicity. So just to conclude in this short presentation, here are projects, and in NanoSolve we use such technology to score over 20 different nanomaterials. In Subdoma, the high throughput is developed even further in a new way, where we have also rapid answers. So it's time and feedback directly. And you heard also from Andrea early today about the Harmless project, where it's the tiered approach is also fully active. So I'll stop there and thank you for the opportunity to present a little bit on the high throughput concepts.
Thank you, Roland. And quite clearly, one of those projects really has a central role in the area of early iterative make test, which I think high throughput and these technologies can really make decision making much quicker and easier in the make test cycle, which is akin to what was used in the pharma industry in my day, of course.